Well, hello everyone. Welcome to APHA's weekly open forum webinar series on COVID-19, addressing the crisis. I'm Michael Hogue. I'm your host, president of APHA and dean and professor at Loma Linda University School of Pharmacy in Loma Linda, California. We're excited that you're here today. We've got a great program for you with a topic that has been so hot in the last few weeks. We're going to discuss antibody testing and how to evaluate antibody tests and really the implications that these tests have for us as pharmacists, because as we all know, we are now authorized to conduct these tests. So it's really critical that we understand this. It's an evolving science, but we've got some experts today who really know in depth a lot about these tests, and we're going to get you the latest and greatest information that we can. Now, our two guests that we have today, first of all, is Dr. Paul Herman. Dr. Herman is a physician, professor and chair of the Department of Pathology and Human Anatomy here at Loma Linda University. Just to add that Dr. Herman has been in charge of validating all of our COVID testing and antibody testing here at the university in our 6 hospitals and throughout our health system, because we need valid results and need to know that they are valid. So he really understands this at a great level of detail, and especially the implementation steps. Pardon me. Our second guest is really no stranger to all of you who've joined us every week. It's Dr. Dan Zlot. Dan is our Vice President of Professional Education Resources at APHA, and Dan has a very long history of working at the NIH as an oncology specialist and working with immunotherapies and immunology. And Dan is really going to lay a foundation for us of understanding today about how COVID-19 creates antibodies in our system. Now, remember, all of our calls are designed to answer your questions about COVID-19, and sometimes your questions go beyond our topic, and that's wonderful. We're happy to have them. Joining us also today then will be ELISA Bernstein, and all of you know Dr. Bernstein as well if you've been joining our weekly webinars. She's our Senior Vice President for Pharmacy Practice and Government Affairs at APHA. And she has some very late breaking news that she's going to be sharing with us later in the webinar. So you'll want to hang on and not leave the webinar, because she's going to have some really great information to share with you, some clarifications from the Department of Health and Human Services regarding pharmacist testing. So lots of great information. Let me share with you some details about today's webinar. Following this brief introduction, we'll have presentations, brief presentations from both Dan Zlot and Paul Herman. We'll intersperse a few questions in there for these gentlemen to answer for us relative to COVID-19. But on the right-hand side of your computer screen is the GoToWebinar control panel, and there's a place for you to be able to submit your questions. Please go ahead and submit your questions now. In fact, I've already got a host of questions that have been submitted. You all have been thinking ahead, and so it's wonderful. Keep adding your questions in as you think of them. We would ask that you please enable your audio control pin. If you've joined us for audio by telephone, you'll have to go ahead and enter your audio control pin so that when I call on you to ask your question, your line will actually unmute. So if you have a question and you don't have audio capability and you want me to ask the question on your behalf, just simply put no audio beside your question, and I'll be sure to ask the question on your behalf. And clearly, I have got to take a drink of water, but we're going to get on with our presentation today. And I want to remind you that there are handouts. Today's slide presentation is available as a handout in the handout tab on GoToMeeting. And in addition to that, we are recording the content today. It'll be available within 24 hours at pharmacists.com on the COVID-19 resources page. Of course, you all will get an email with that information as well. So without any further ado, I'm going to turn it over now to Dan Zlot, who's going to kick us off with an overview of COVID-19 infection and our immune response. Dan, activate your webcam, please, and we'll go from there.
All right. Well, good afternoon or good morning, everyone. Very nice to speak with you all. So I'm going to kick off just by giving us a little bit of an overview of a COVID-19 virus and then a little bit about how antibodies are developed, and then I'll turn it over to Dr. Herman. So just really quickly, the COVID-19 disease is caused by the severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. It's certainly a mouthful, so just for the sake of simplicity, we'll call it the COVID-19 virus, and we'll use that term interchangeably. So the COVID-19 virus, I feel like all of us have been getting a PhD in virology throughout this whole experience, but the COVID-19 virus is an enveloped RNA virus. So as it buds out of the Golgi apparatus, it steals a little bit of the Golgi membrane and wraps itself in a lipoprotein membrane, which is a little bit different than some other viruses. And it's an RNA virus, so it has RNA and not DNA. And it consists of a couple of different layers. So on the outside are the spike proteins. And so on the image there, you can see those little red dots studded around the virus. So those are the spike proteins, and those are what allow the virus to actually get into the cell. So those spike proteins interact with ACE2, angiotensin converting enzyme 2, that's present on a number of different tissues. And that's how the virus gains entry into the cells. So you'll also hear that referred to as the S protein. Then there's the envelope that we talked about. And within the envelope are a couple of different proteins, the envelope proteins as well as membrane proteins. And those are somewhat similar. They provide structure to the virus. They link the envelope to the capsid, things like that. And then inside of that is the nucleocapsid. And the nucleocapsid has the nucleoproteins or N proteins. And then inside of that is all the genetic material, which is the RNA. So those are sort of all the different layers of the virus as you go from the outside towards the inside core of the virus. In addition, there are a couple of other proteins that are associated with the virus, and those are referred to as non-structural proteins, or NSPs. And then there are additional accessory proteins, and they come from the open reading frames of the viral genetic code. So those are areas where the cellular machinery can kind of sit down and go along the genetic material and then create proteins. So those proteins are often called open reading frame proteins, or ORF proteins, as well as others. So the reason we're talking about all the proteins is we're going to talk a little bit about antigens that can potentially generate immune responses. So next slide, please. So when it comes to the actual COVID-19 antigens, there are actually a huge number of potential epitopes that have been confirmed or that are hypothesized to generate an immune response. So that we're very fortunate. We live in an amazing age. There are some amazing databases that let us screen all these different proteins, amino acid sequences, et cetera, against these databases to see if similar structures have been recognized by the immune system, and if so, that gives us a pretty good idea that there's a strong likelihood that you can generate an immune response against a particular amino acid sequence, glycoprotein, et cetera. So when it comes to the COVID-19 virus, it turns out that the most antigenic proteins are found probably on the S protein, followed closely by the N protein, and then the M protein. So the S protein, again, is a spike protein, the N protein is the nucleoprotein, and the M protein are the membrane proteins. In addition, the non-structural protein 3 and non-structural protein 4 have been identified, as well as open reading frame protein 3A and 8A. So I bring all these up. In the future, we may talk about vaccine development, and these are all potential targets for vaccines that have been hypothesized or confirmed. Additionally, T cells and B cells play an important role in developing an antibody response, and we'll talk about that in a second. But these same epitopes have also been demonstrated to generate both T cell and B cell responses. Now, for the sake of antibodies, we're going to be focusing mainly on B cell responses, but just to kind of give you an idea about how important these are. Now, interestingly enough, the image to the left is actually a scanning electron micrograph of the COVID-19 virus, and those little green dots around the outside of the viruses, those are the spike proteins, so you can clearly see those on electron microscopy. Next slide, please. All right, so now let's move from the virus itself into a general review of antibody development. So as we all know, antibodies are produced by activated B cells, which are also called plasma cells. B cells recognize their targets through an immunoglobulin that's present on the cell surface, which is also referred to as the B cell receptor. The B cell receptor is, well, each B cell has a very specific B cell receptor, and it also makes, if it were to make antibody, it makes one specific type of antibody. And so that's the first step in B cell activation. Now, once a B cell recognizes its target, it must then also be activated by a CD4 T cell. So once the B cell recognizes its target, it sucks that target in, digests it, and presents it on the cell surface. The T cell comes along and binds to that presented antigen, and then if it's activated, that T cell will then start to secrete B cell activating factors, and those are things like interleukins, IL-6, for example. And then that will then activate the B cell. Now, activated B cells typically take one of two paths. They can become a plasma cell and start to secrete antibody, or they can become a memory B cell. And we'll talk about the function of memory B cells in just a little bit. But those help us develop that long-term response and mount an immune response more quickly when we get re-exposed to a pathogen. Now, each plasma cell makes a single type of antibody and a single class of antibody. So B cells can make IgA, IgG, IgE, IgM, or IgG. They will only make one of those. And of that class, the antibody that they make will only recognize one specific antigen. So that's sort of how they work. They're very, very specific. Next slide, please. So that's an overview of the different types of antibodies and how B cells work. Now let's talk a little bit about timing. And this, of course, will be really important as we start talking about testing and how to interpret testing results. So as you are probably aware, from the time you get exposed to a virus, it takes a little bit of time to develop an antibody response. So traditionally, we think of IgM as sort of the initial antibody produced when we're exposed to an antigen or a pathogen for the first time. And that takes about 4 to 7 days after the initial pathogen exposure. The IgM levels will rise for about 7 to 10 days. They peak there, they'll plateau for a little bit. And as you can see in the graph, they start to fall off between 2 to 3 weeks after initial exposure. And then next comes IgG. And so IgG is what we think of as our longer term antibody. And the reason that's thought that way is because when we're exposed a second time, IgG is predominantly produced rather than IgM, and it provides sort of our long-term protection upon a second exposure to a given pathogen. Now, IgG typically takes a little bit longer to develop. We think of about 7 to 10 days after initial exposure to a pathogen. It peaks about 10 to 14 days after initial exposure, so a little bit longer. And then, as you can see, it hangs around for a little bit longer as well. You know, upwards of 2 months sometimes before you start to see that plateau start to drop back down. So, as you can imagine, there's some potential implications there depending on when in that time frame you get tested for antibodies. If you get tested really early, there may not be much of a signal. If you get tested late, you might only see IgG and not IgM. So this information is really important in terms of how we interpret those tests. And then, as I mentioned before, the memory B cells are really important for us upon re-exposure. They're sort of our early warning system, and once they see a pathogen that they recognize, they rapidly go and reactivate plasma cells so that we can produce lots of IgG much more quickly than we did initially, and that helps prevent reinfection. Next slide, please. So now that we've talked about the general process of antibody development, the big question that everybody's been asking is, do people actually generate antibodies to COVID-19? And the overwhelming answer is yes, absolutely. So this was a cross-sectional study from 3 hospitals in 285 patients. And what they found in this study was that 100% of patients developed either IgM or IgG, or even both, against COVID-19 within 19 days of symptom onset. Now, it's important to understand there's a little bit of a delay between viral exposure and symptom onset. For COVID-19, that's anywhere between 2 to 14 days. That's sort of the incubation period of the virus. But the overwhelming majority of people will develop an antibody response to COVID-19. That's been confirmed in a couple of other studies as well. And all patients in this study were confirmed to be COVID-19 positive by a PCR test, and then the serum immunoglobulin levels were tested by ELISA, or enzyme-linked immunosorbent assays, and Dr. Herman will review those for us a little bit later on to describe how they work. So hopefully that sets us up nicely to understand a little bit more detail about how these tests work. So Michael, I'll hand it back over to you.
Dan, thanks for that. That is an excellent overview. Before we run into Dr. Herman's presentation, I do have a couple of questions that have come in for you, one that came in ahead of time and one that we just received in the question box. The first question is, if an antibody test is positive, is there a risk that a patient could be a carrier of the virus and spread it? This is a concept that's frequent. We hear this with other infectious diseases as well. So is it possible to carry the virus even if you're antibody positive?
Yeah, that's a great question. And so the answer is, I think, definitely yes. And I'll get into a couple different scenarios as to how that could happen. So if you have antibodies, that does not necessarily indicate that you've cleared the virus. So when you look at the antibody response curve in the chart, we saw that people started developing antibodies as soon as 2 to 3 days after symptom onset. At the same time, when we look at the data about PCR positivity and the persistence of COVID-19 PCR positivity, people are positive for an average of 20 days based on the data coming out of China. So if we make the assumption, and it is a little bit of an assumption, that if you have positive PCR test, you're infectious, then there's a period of overlap there. So certainly yes. Now, another aspect of this is, even if you, let's say you're not infected per se, you don't have the virus in your nose, in your mouth, oropharynx, etc. But let's say you work in an environment where there's lots of COVID-19 around, like a pharmacy or a hospital. It's really important to understand the concept that, we can bring it with us, not in our nose or mouth, but on things, hands, shoes, clothes, things like that. And so that's another way that we can become carriers, not in the sense that we have it in our system, but we can bring it with us and potentially expose family members. So
the key,
a couple different ways.
The key here is that if you have a positive antibody test, don't let that build your confidence to think that you can't potentially infect someone else, because it's not always just about the respiratory secretions. It could be
Absolutely.
because of your work environment. So that's a great point. One more question, and then we're going to move to Dr. Herman. Mary Gibson, one of our participants on the call, asked a question but doesn't have audio. And her question really is about thinking about getting tested. Maybe 2 or 3 months ago, had a flu-like illness, loss of taste and smell, never did get tested for COVID because the test wasn't available. Is it reasonable for someone like Mary to get tested with antibody testing now? Is that a reasonable thing to do?
Yeah, I think so, for sure. If there's tests available in your area, I think as a healthcare provider who's on the front lines, I think there's a level of reassurance that's provided by knowing that you've potentially been exposed and developed, hopefully, some level of immunity against the virus. And so, not that that changes necessarily the way we need to behave or how we protect ourselves, but certainly, as a pharmacist myself who works in hospitals, I would feel much more comforted, if I was working in an ICU or if I was seeing patients face to face, if I knew that I'd been exposed and maybe was on the other side of this and had some degree of immunity. So if that would provide comfort to you, then I would say there's definitely some good benefit to being tested.
That's great. Well, Dan, thanks for that. We'll come back to you for more audience questions later. Dr. Herman, welcome. And if you'll activate your webcam, we are anxious to hear your presentation.
All right. Thank you, Michael. The first slide here shows the ELISA, which stands for Enzyme-Linked Immunosorbent Assay. And this particular assay takes advantage of the antibodies that Dan introduced us to very nicely. So if you start on the left side of the screen, there's a picture of kind of a green triform object. There's a little green circle sitting down on a white line. So that little green ball represents the antigen that we were attempting to test for. The green sort of stick figure there is an antibody that's been made in a laboratory against it, And at the top of the little green stick is an orange ball, and that corresponds to the enzyme. If you look then, the enzyme, the orange ball, there's a tiny little red arrow, very faint, that goes over to a red ball, and that's indicating that substrate has been converted by that enzyme in the orange ball into something that's measurable. So we call it an enzyme-linked immunosorbent assay because the antigen has been absorbed by an antibody. That antibody is attached to an enzyme. The enzyme changes a substrate into something that can be measured with a spectrophotometer because it either fluoresces or has a color associated with it. Along this graph are 4 different types of ELISA, or enzyme-linked immunosorbent assays. The first one is a direct. The second one is what we call an indirect ELISA. This one has an antibody initially binding to the antigen, and then a second antibody attached to the enzyme binds to the back of the primary antibody. The reason companies do this is it allows them to make a single signaling antibody against the back end of a standard antibody, and then they can vary the standard antibody for all the different antigens that they wish to look for. And then there are 2 other examples. There's the sandwich assay, which is shown second from right, and then on the far right side is a competitive ELISA assay. But for the purposes of today, I think looking at the first one is probably adequate of just having an antigen. There's an antibody attached to an enzyme. The enzyme converts a substrate into something that can be seen or measured. So this is the ELISA test for antibody. On the next slide, we have the lateral flow test. So the lateral flow test looks a lot like a pregnancy test kit. You can see a picture of a real one sitting there on the right with a black box covering the patient's information. That's one we actually used in the lab. On the left, let's start with the cartoon first, is a cutaway of it, and you see that there's a little sample pad that sits underneath the circle. The patient's serum is dropped into that spot. Within the serum, there are antigen analytes. Those antigen analytes then float through a chromatography process into the conjugate pad that's shown in green that has on it some antibodies attached to nanoparticles. So the antigen moves up into the pad, and all the antibodies on that pad, some of them may attach to antigen if there's antigen present, or they may not. And the whole thing floats forward until it comes to those 2 red lines where it has antibody labeled. The first red line has an antibody that is targeting one of the particular antigens that was in the serum. And so if the antigen was present, then the antigen will now be bound by the antibody that's in the red test strip, as well as the antibody that carried along the nanoparticle, and it will stop there. If there is no antigen present, then the antibody will have no reason to stop at that first line. It will move to the second line where the antibody will stick directly to the antibody that was in the conjugate pad. And so this is the way you get a line that shows if there's antigen present, and then followed by a second line that's the control that demonstrates the test actually worked. So if we look at the image on the right there, you'll see a very faint line right next, slightly below the letter M in the window panel there. And then if you look up higher, there's a C for control and then a much darker band. So this particular test was positive for the IgM antibody, indicating the patient had IgM present within their serum. And as Dan mentioned, that's the first antibody that we expect to see in an immune response. Okay, next slide, please. So when we get a new test in a lab, it's very important that it be validated, and we'll come back to this point in a few minutes. Validation is a process whereby the laboratory that's running the test, by law and also by good practice, has to demonstrate that the test is precise, accurate, reproducible. You have to determine the minimum limit of detection, or the range over which the test is valid. You need to correlate that test to an existing method when at all possible. And then finally, you have to determine a relevant reference range for the test. This correlation to existing method, relevant reference range, and limit of detection is what made testing sort of come along so slow relative to the outbreak of this epidemic, because there wasn't a lot of prior experience with it, and so finding things to correlate with were very difficult. Knowing how to determine a limit of detection when there's no standard available is almost impossible, and determining a relevant reference range, as you might imagine, if you can't do those first two things easily, the next one's going to be even more difficult. This is the reason that testing has been a little bit problematic, as we've seen in the news, and it's also the reason the FDA went for the emergency use authorization model, rather than insisting that the tests be fully validated before releasing into the market. If we could move to the next slide, the data there show what we did when we first started working on a lateral flow immunoglobulin test, and that's the test that you saw the example of in the earlier slide. In order to do valid antibody testing, you have to have some idea of the patient's physiology. And this chart is a chart we made of a number of patients that we knew had COVID because we had tested them with the virus and we knew it was present. And so the first thing we did with our test was study them over a number of days until we could determine the approximate time range after viral testing that we would start to see a response with IgM and a response with IgG. And so this particular graph shows what Dan presented earlier, that first you have no response shown by the orange boxes, and as you go further you get an IgM response. As you progress further in time you'll see IgM and IgG, and then finally if you wait long enough you'll get IgG only. Once we had this graph, we were then able to go back and get sensitivity and specificity determinations for the particular antibody test that we had purchased. So on the next slide, you can see those particular tables that were constructed. Based on the slide I just showed you, looking at the patient's physiology over time, we decided to have a sensitivity for within 5 days of a positive PCR-based test and a second sensitivity within 14 days of a positive PCR test. And as expected, the sensitivity of our test, meaning either IgM, IgG, or both, would be present within 5 days of a positive PCR test, was about 75%. And if we went out the full 14 days for the particular test cases we looked at, it approached 100%. So this is very important to know when a clinician or other healthcare provider tries to interpret the test, is having some understanding of the opportunity for false negative as well as the time frame of the disease. On the next slide, we'll start to talk a little bit about the regulatory climate, which I alluded to earlier. Ordinarily, in the laboratory world, a test is developed by a corporation, and the FDA then evaluates and approves it after doing some extensive testing. After the FDA has approved it, the clinical labs can then do a very quick validation to make sure everything's working correctly and they can turn it on. Due to the current situation, as I alluded to earlier, the FDA was in a more difficult situation and they issued emergency use authorizations, which initially said only high complexity CLIA certified labs were allowed to do the testing, which sounds pretty good at first until you realize that those labs just inherited all the work that the FDA would ordinarily have performed, which made it a lot more difficult to bring up the testing. Antibody testing, unlike the viral testing, was turned loose by the FDA even before EUAs were fully issued. So companies that had applied for an EUA could go ahead and market the test. What this means is it became even more important that the labs do very thorough validations, because without any backing from the FDA, they had to be absolutely sure they knew what the test was doing and how it worked. If we could go to the next slide, please. What that means is it's extremely important, if you want to offer a test, that you find a good test. So one option would be to collect the specimen and partner with the lab that has validated their testing. Make sure, if you do that, that the lab is reputable, and I would ask to see their validation data. If, on the other hand, one wishes to go it alone and bring in their own test within a lab, I would highly suggest they go to the web page that's shown here. If you type in the Google search that's listed there, COVID-19 serology test and FDA EUA, you'll go to one of the choices that you see there on the left side, EUA authorized serology test performance FDA. If you click on that, you'll get a list of the testing characteristics and performance of all the serology tests that the FDA have currently issued authorizations for. And each one of them, and I blocked out the name of the particular company here in fairness, because I understand this is not supposed to be a biased presentation, but if you look at each one of the tests individually, it will have the data shown there, which gives you the sensitivity and specificity for IgM, IgG, the particular test performance characteristics, and you can select the one that you believe would meet your needs to the best ability. There's also some contact information, which can be very helpful if you're trying to figure out what test to bring in. The FDA is generally quite helpful, although I will tell you, you may have to wait through a phone tree for a while to get to someone, but they're interested in helping you, and I would urge anybody who's thinking of setting up testing to take advantage of that. I believe the next slide is my final slide, and it shows the timeline for bringing a lab test in. The only reason I suggest that you take a look at it is some experiences I've had here where people assumed once a test was available on the market, they could start ordering it, and you don't want to be in a situation where you're either turning away patients or trying to collect specimens while you build something. So if you want to bring in a new test, recognize there's going to take a certain amount of time to buy it. Once it's there, you're going to have to install it, validate it, determine when you want to go live, and then of course good practice is to continue to monitor the test characteristics while it's running. Don't forget that it's also very important to make sure supply chain is in place and that the supply chain is robust, because you don't want to be running testing and run out of supplies, as we saw demonstrated in the last couple of days in the news that came out of Florida, where somebody had partnered with a lab, and unfortunately the lab had apparently run out of supplies but hadn't told anybody, and it was a huge debacle. So that concludes the formal part of my presentation, and thank you very much, Michael.
Well, thanks, Paul. We've got a couple of questions that have already come in. Just to give everybody the ground rules, when I call on an audience member to present, the staff will unmute your line, and you have up to 60 seconds to state your question so that we can take as many questions as possible. This first question is pharmacist Debras Weiss, W-E-I-S-S. We're going to unmute your line and allow you to ask your question, Deborah. Okay, I'll ask Deborah's question for her. The question is, if one develops antibodies, are we to expect that the individual would have those antibodies present for life? In other words, if a person gets tested 10 years later, will that person still show that they have antibodies? And do we have any understanding of what antibody level is a correlate of protection against disease?
Well, nothing like starting off with a really good question. Not sure I have such a great answer, but I'll give you my best assessment. As Dan showed very nicely, in standard immune response, the first thing to come along is an immunoglobulin M antibody, which will last for some number of days to maybe a couple of weeks, give or take. And partway through that, immunoglobulin G will start to rise and will stay elevated for a longer period of time. It depends on the particular virus and disease as to how long that immunoglobulin G stays present in the person's system. You may have seen in the news recently a lot of chatter about the fact that having an antibody may not provide protection long-term against COVID-19. And the reason those statements are being made is there are some very rare examples of other coronaviruses that triggered an immune response in an individual, but over time that immune response went away to the point that it didn't appear to confer any additional immunity. Nobody knows what the situation is yet with COVID-19. It's going to take some time to find out. In terms of how much antibody you need, I don't know that there's any clear demarcation that anyone's aware of as to what level of antibody presence one would need to have some kind of immunity. My personal opinion, and this is strictly opinion based on sort of a gut feeling of a lot of literature, is if a person has seen a particular entity before, I believe they'll have at least some immune response to it. It doesn't mean they won't get sick again, but it is possible that if they did get sick, the sickness might be somewhat attenuated relative to the last round with it. But until time has lapsed for probably a half decade or a decade, it's going to be impossible to really say how much protection our immune system gives us on a regular basis to the COVID-19 or the SARS-CoV-2 virus.
Right. That's great information and good to know. Now, I'm curious, Paul, as a physician, what would you expect of a community pharmacist who decides to conduct antibody testing at their pharmacy? First of all, do you support community pharmacists doing antibody testing as a physician? And then what would you expect that they do if they're going to do it?
Yeah, let me be clear. I do, in fact, support it. I think that it is very useful. I think the pharmacist in some ways may be in a better position than the physician to offer the kind of testing that we need in this country to open it back up and to make good use of quarantining care and all the kinds of things that we're going to have to sort through. So I'm a strong supporter of pharmacists performing it. The expectation from my perspective would be the patient needs to get a very clear report as to their test. It needs to tell them that it was positive or negative. It needs to give them some indication of what that means to them. So a little bit of education around the timing of serology, you know, when the IgM would come along, when the IgG would come along, and a very clear explanation of what the test can and can't do, including the particular pitfalls of that particular test. Depending on the level of interest of the patient, maybe going so far as to talk a little bit about the sensitivity and specificity. I think that's kind of the bare minimum. Beyond that, though, probably depends a lot on the relationship between the pharmacist and the physician within the particular community where they work. In a major city, I would say that's probably all that's needed and all that could be expected. I suspect in some smaller communities, we may have situations where there's a very tight connection between the healthcare community among nursing, physician, pharmacists, dentists. In that community, I would expect that there might be some crosstalk between some of those professionals, both referring patients and possibly the healthcare providers in the clinical setting asking the pharmacist's advice in terms of what the test means and helping guide therapy and care and the like. So it's going to have to titrate a little bit to the community, but the bottom line is anybody performing testing needs to be able to educate the patient fairly clearly and succinctly as to what it meant for them.
Okay, great, good advice, and that's good information. Dan Slott, how about you activating your camera and joining us here together, and we'll do some back and forth questions. I will take the next question from Dr. Theis. I don't know if this is Brad Theis or which Theis that is on the line, but Pharmacist Theis, we'll unmute your line.
Can you hear me, Michael?
There you go. It is Brad Theis. Okay, Brad, go ahead. I can hear you now.
Hey, I just had a question about, we're seeing a lot of labs develop their own tests, LDTs. That's like pharmacy compounding. Do labs that develop the test under an LDT authorization have to have an EUA, or can they also offer the test through that LDT validation if they can? And can someone be comfortable in working with them if they can provide their own validation data?
Dr. Herman, I think that's your question. Yeah.
Sure. Be happy to tackle it. The LDT arena, in light of the EUA, has been a little bit of a fuzzy moving target. So in the absence of an EUA mechanism, if you go back, before COVID and when there weren't very many EUAs, every lab in the country, I believe, had some laboratory developed tests that they had to develop locally. Your validation method, obviously, in that situation has to be absolutely scrupulously rigorous. I mean, you got to go beyond just what I described here, because it is very important that you know what you're doing and you're able to explain what you're doing. And I can assure you there are inspections that come around routinely between the Fed and state licensing bodies that will ask to see that. So it was regulated in that environment. There were some problems initially that were noted around that during the Theranos debacle. I don't know if any of you remember that story. It was the startup company in the Bay Area. At that point, the FDA started to take a little bit more closer scrutiny of laboratory developed tests, but they were still permitted, and it really didn't change all that much within the industry. I think there was a little bit more paperwork that laboratories had to file. With the COVID-19 epidemic and this new mechanism of EUA, I don't know that it was a new mechanism, but the lab world hadn't dealt with it very much. But the emergency use authorization situation, the FDA initially with the viral testing took the tack that if you had a laboratory developed tests that you were going to be offering, you needed to submit that for an EUA by the FDA. And so if you look at the EUAs that have been issued by the FDA, the very early ones are for the CDC lab, I think the WHO lab, and there was one in the New York Public Health System. And then University of Washington got one, and then we started to get more commercial providers offering testing in this arena. Serology has taken a little bit different path, which has been kind of interesting. When serology testing first came along, the FDA wasn't as concerned about it initially as it had been about the viral testing because it wasn't viewed as definitive, because everybody knew there was a lag between when someone got infected and when the virus came up. The FDA said that, you know, it would be fine. And so there are a lot of laboratory developed tests in that arena. Then the FDA went a little farther and said that companies were free to sell their testing as vendors to other laboratories as long as they had notified the FDA and were submitting an EUA. And at that point, there were a lot of people who came out of the woodwork because they could start selling something as soon as they'd sent a letter to the FDA. About a week and a half ago, the FDA kind of started shutting that down, and appropriately so, because anytime you get in that environment, there are people who take advantage of it. So again, my advice now, with the number of tests that are listed for serology and viral testing on the FDA website holding EUAs, I would recommend anybody wanting to get into it probably would be best served to try and pick from one of those that's listed under emergency use authorization. I don't know if that gets at the heart of your question, but it is a little bit of a nebulous arena right now.
Great. Okay, we're going to take a quick question from pharmacist and Professor Anandi Law. Dr. Law, we're going to unmute your line.
Hi. Can you hear me?
Yes, we can hear you.
Okay, hi. Thank you. And my question is really quick, and it might have been addressed already, but you mentioned, I think it was Dr. Zlot who mentioned that the IgG kind of tapers off over time too. So I just wanted to know how long is that anticipated? So how long is the immunity in the body? And I think I just saw a study today which says there is some immunity, but I just don't know how long it is. Thank you.
Dan,
Sure.
go ahead.
Yeah, that's a great question, Anandi. So I'll start off with what we would typically expect, and then I'll finish off with the answer to COVID-19. So in typical diseases or things that we're vaccinated against, you would expect to see the IgG levels taper off, but still be detectable. So if you're familiar with the concept of antibody titers for vaccinations to see if someone needs to be revaccinated against a particular disease, that's because there are constant low level levels of IgG present against that particular antigen. And so if COVID-19, and this is a big if, we don't know this yet, follows the traditional pattern, we would expect to see something similar. Now, the big question, as Dr. Herman alluded to, is how long does the immunity last? Is it 5 years, 10 years, 20 years, a lifetime? And that we don't know yet. And as Dr. Herman said, I think we'll have to wait, unfortunately, 5, 10, 15, or 20 years to fully understand the answer to that question.
All right. Well, we have gotten, folks, just a ton of questions. This is clearly a hot topic. All of you are very interested in it, and we will attempt to answer some questions that we don't get to today on the Engage platform in the coming week. But I'm going to take our last verbal question before we move into some additional information. Pharmacist Anthony Pudlow, we're going to unmute your line so that you can ask your question.
Oh, can you guys hear me?
We can hear you.
Oh, great. I just know we've been hearing from a lot of just a lot of concern over some of the diagnostic testing and the validity of them, you know, focusing on false positives. I know you spoke about the validity and the sensitivities to the serology test. Is the FDA, I haven't been able to find a lot of great information on it, but I just know there's some concerns right now about diagnostic testing, especially as we see pharmacies, more and more community pharmacies doing the diagnostic tests.
All right, Dr. Herman, let's let you give the first response on this one.
Okay. That's actually a very interesting subject right now. It's sort of hot in the news. We've seen some interesting news articles in the last 2 weeks related particularly to a vendor that's used in the White House testing. I don't think the story's completely told yet. My sense is that continued following of that story would be well worth one's time. But I do think as a person starts to think about wanting to do testing, regardless of their background, pharmacy or otherwise, they need to think a little bit about the possible places where errors can come into the testing. False positives with a nucleic acid-based test generally occur when there was contamination. It's not particularly common in that arena to get a false positive from some other nuclear material, assuming the test was designed very carefully around the actual genes within the COVID virus. False negatives are much more common, and they come about for 2 reasons. One is, no matter how good the testing equipment in the laboratory, it's going to require some number of viral particles greater than 1, quite a bit greater than 1 in most cases, to turn it positive. So there's going to be what we call the analytical sensitivity, or the analytical limit of detection, meaning there have to be enough viral particles in the specimen for the test to actually notice them and behave appropriately. Analytical sensitivities are never 100%. My guess is the best you can hope for is probably about 97, 98% in the analytic arena alone. But now let's talk about testing the way it gets used in the field. One starts by inserting some sort of collection device into the nose or airway. They get the virus on that, and then they put that into a solution, and then the solution goes to the lab, and the lab puts it in the machine, and the machine tests it. The inability to guarantee that that swab is collecting a virus every time means there's going to be some decreased sensitivity just from the collection methodology. Multiply that by the analytical number that I pointed out, and what it means is you're always going to have some false negatives. The question is how many, and that's where the story is going to be most interesting, I think, as it unfolds around the current testing paradigm. It plagues everyone. I don't think pharmacists are in any more danger than anyone else performing the testing. It's just one has to keep that in mind.
Okay, well, we do have some information that we want to have ELISA Bernstein share with us, but we've gotten a lot of questions on one subject, and I'm going to ask if one of you guys could just give us a very brief answer to this question. And the question is this: how frequently should someone be tested? So, for example, community pharmacies have largely closed their lobbies, and there's a lot of discussion about that there needs to be increased testing available. Should pharmacist owners and pharmacy managers and corporate owners, should they be regularly testing their staff with antibody tests or with COVID-19 tests, and how frequently should they be doing that testing? Any suggestions, ideas? Is there any guidance out there on this, or is this just right now an unknown thing?
Well, I'd be interested in hearing all the opinions. My view is it's going to probably be pretty much person dependent, the answer you get. My bias would be anytime you have a sense of symptoms, it would be worth getting tested. Anytime the testing is going to change something, either whether you're going to go in the hospital or you're going to sequester, it's worth doing. If you had a staff that was in a highly exposed environment, it might make sense to have some sort of cycled frequency, but I wouldn't guess to know what. I'd be interested in your view, Dan.
Yeah, I think they fall very much along the same lines. I think it depends on what you're doing at your particular place of work, and then also which particular test you're talking about. If you're talking about antibody testing, once you've got positive antibodies, I don't think there's any further benefit to doing sequential antibody testing. If you've got them, you've got them. Whether it's worth doing a PCR test after antibodies, I think that's certainly up for debate. Probably not a bad idea in this acute period here, just to make sure that you're not a carrier and potentially putting others at risk. If you're completely negative, then I would agree with you. I would say it's based on symptoms and when things would change. So I think that's kind of my general 2 cents.
Good. Well, Dan and Paul, thank you so much for joining us. We'll let you mute your cameras. We're going to ask our audience real quickly a polling question before we share some HHS data with you. My pharmacy practice site is considering offering antibody testing when it becomes available. Yes, no, or not applicable to my practice. We're going to give this 20 seconds so that everybody has a chance to vote and just getting a pulse here. So please respond quickly. Just click on your screen. Yes, no, or not applicable to my practice. All right, Brian, let's get the results, or Mitch. Looks like about 36% of you say yes, that you do plan to do antibody testing when it becomes available. Okay, now I want to turn the floor over to ELISA Bernstein, who has some really important information to share regarding some guidance that came from HHS this week. ELISA, the floor is yours.
Great, thank you, Michael, and thank you, Paul and Dan. That was really clear and informative. I really appreciate that, and I'm sure our listeners did as well. What I want to share with you is something that came out 2 days ago from the U.S. Health and Human Services. They announced an interpretation that they put out that has broad impact on the ability of pharmacists to order and administer tests across the country. They concluded that licensed pharmacists may order and administer COVID-19 tests even in states that prohibit licensed pharmacists from ordering and administering these tests. This means that in your state, if your state cannot limit or prevent pharmacists from ordering and administering COVID-19 tests. And this confirms the administration's intent and confidence in us as pharmacists to provide expanded and widespread testing capacity for Americans across the entire country. What this does is it follows an April 8th, 2020 guidance that the Secretary issued authorizing licensed pharmacists to order and administer COVID-19 tests, including serology tests that FDA has authorized. And so we've seen that some states have issued executive orders and declarations or clarified, what licensed pharmacists can or can't do. Other states have limited or prohibited pharmacists from performing these tests. This provides clarity. Pharmacists still need CLIA certificate of waivers to perform the point of care tests that FDA has authorized, and we're still assessing how this impacts state CLIA requirements. The opinion only speaks to COVID-19 tests that FDA has authorized, and FDA's website has a very clear chart that indicates which tests are authorized for point of care, which includes pharmacies. It doesn't address flu or other tests, and it doesn't address immunization. So this also doesn't solve the payment and billing issues, and so we're still seeking clarity on the payment pathways for all patients, particularly Medicare patients, and when you are testing versus just collecting specimens and sending that to a lab. So APHA, and along with our pharmacy association partners, we've been meeting with CMS and congressional staff to get clarity and make these pathways more clear, predictable, and financially supportable for you as a business model. So we're on it, and stay tuned for more information. If you could go to the next slide. One way to get clarity is to have Congress be more clear and issue a law that would enable pharmacists to be providers under the law for testing for Medicare Part B patients. So we want you to take action. Go to the actioncenter.pharmacist.com, and in one very easy click, you can send letters to your legislators to tell them to make this happen. Next slide. Oh, this is Dan. You want to talk about, or should I?
Actually, I think I'll pick it up here. And
okay, thank you.
Thank you, ELISA, for that great information. We do want to remind everybody that each week, Dan Slott and his team in education release a continuing education series called APHA's 15 on COVID-19. These are 15 to 20-minute CEs. There are 2 new episodes, one on anticoagulation in COVID-19 and one that's similar to today's topic that gives you a little additional information on antibody development and reinfection, and we had some questions about that today. Other thing we want to share with you is that there's a variety of resources that APHA has available, practical information that we keep up to date, and we would encourage you to go to pharmacists.com, click on the COVID-19 link. Very important information about fraudulent medical products and scams, and I think it's very relevant to the topic we discussed today relative to antibody testing. Pay very close attention to this resource, and we would encourage you to take a look at this. There's even something about vaccine development in one of our resources there. Now, we do this every week, but we also want to make sure that there's other opportunities for you to engage, and that's through pharmacist.com's Engage platform. And you can post questions, support your colleagues, share lessons learned, and each week we do pick 1 or 2 of the questions that we did not have a time to get to in our webinar, and we have an expert answer those questions in the Engage platform. So please pay close attention to that and follow that discussion. It's a great way for you to stay engaged with this conversation. Now, next week, we will be doing this again on Thursday, May the 28th at the same time in the same place. Our topic next week will deal with 2 subjects. We're going to discuss a little bit more about the details of payment for testing. Dr. Bernstein shared with you the details of HHS's clarifications to this point. Next week, we're going to bring you a little bit more detail about payment. We think that'll be a valuable discussion for you. And we're going to address a topic that has been very, very important and on many people's minds. Pharmacists are immunization hubs in their local community, providing adult and adolescent immunization for a variety and host of diseases. And of course, there's been lots of questions about immunization during COVID-19. And next week, we're going to address your concerns and questions about providing immunization during the COVID-19 pandemic, and may even get into a little bit about COVID-19 vaccines. So it's going to be a chock full week next week. You won't want to miss it. Be sure to share this with your friends. The link for registration will come out tomorrow, and we encourage you to register for that. For today, I just want to thank each and every one of you for what you do to protect your local communities against COVID-19. You're on the front lines, you're running toward the fire, as some people say. As frontline providers, you've been open in your local communities, you've been working actively in your hospitals to make sure that patients receive the best possible care. We at APHA are so proud of our pharmacists, pharmacy technicians, and student pharmacists, and we can't thank you enough on behalf of the profession and on behalf of the patients and communities you serve for all of the work that you do. Thank you, and God bless. Have a great day.